Good day once again and welcome to Naija Pikin. Do not forget to like and hit the subscribe button. On our first video, we talked about how the innate immune response is able to attack invading viral peptide or invading viral particles in the body. And we told you there are two different systems that are able to fight this virus, the innate immune system. Check out the last video on the innate immune system. Today we're going to be talking about the adaptive immune system. Let's take a little recap. I told you when in the innate immune response, it lasts for 2 weeks. The infected cell infected by this virus becomes in a stress state. It becomes in a stress state, and when a cell is in a stress state, it tries as much, it expresses stress receptors on the body of the infected cell, HLA-E, MIC-A, and MIC-B, and there's this binding of these particular infected cells to the NK cells, which are the first defense mechanism that are released by the innate immune response against virus infection or viral infection, as the case may be. So by so doing this, we said everything that happens in the innate immune response lasts for 2 weeks. But how does this particular movement of the innate immune response tend to ignite or activate the adaptive immune system? So we are now going to be talking about the adaptive immune system. Okay, let's say the same virus happen to infect a macrophage. Okay. So I'm going to represent empty theta as my macrophage. Let's say the same virus infects a macrophage. Mind you, on our last video, I told you that every nucleated cell have MHC1 on the body of the cell membrane. But there are other cells that have both MHC class 1 and MHC class 2. An example is the macrophage. So there are cells called antigen presenting cells. These cells are able to have MHC1 and MHC2 on the body of their cells. So I'm going to write them down for you so that you know these antigen presenting cells. APCs, they are called APCs, antigen presenting cells. They are able to present antigen so that they are recognized. Antigen presenting cells. So examples of these antigen presenting cells, we have dendritic cells, follicular dendritic cells, F for follicular dendritic cells. We have macrophages, and we have B cells. These are the types of antigen presenting cells we have. We have dendritic cells, follicular dendritic cells, macrophages, and B cells. For nucleated cells, we said every nucleated cell have MHC1 expressed from the cell. But for antigen presenting cells, they have both MHC1 and MHC2. Thank you. So after knowing this, let's go back to the diagram. So when this particular virus, the same virus, when this virus infects a macrophage, which is an antigen presenting cell, so now we're going to represent our MHC1 and 2 because it's an antigen presenting cell. MHC1 and MHC2. So when it infects the macrophage, so now I have my viral peptide in the macrophage. So the macrophage will be able to express the viral peptide on the body of both the MHC1 and the MHC2. They are antigen presenting cell. They will present this viral peptide outside the body by using the MHC1, so that other B cells or T cells can be able to recognize this particular peptide and do the killing. So they are now presenting the antigen on the MHC1 and MHC2. So you now know the function of MHC1 and MHC2. They are histocompatibility complexes that are able to represent viral peptide or pathogenic substances outside the body of the cell, so that they can be able to present this antigen to whomsoever, whichever cell can be able to do the killing, and also help them in taking care of the virus in the body or whichever pathogen we're talking about. So when the virus infects the macrophage, as you can see, the macrophage here is inactive. The macrophage is inactive. What we want to do here is activating a cytotoxic T cell, CD8 cell. We want to activate a cytotoxic T cell, cytotoxic T cell. But before we can do this, we need a helper T cell to activate the macrophage, so that the macrophage can activate the CD8 cell to produce cytotoxic T cell that will do the killing. That is the idea. That is the idea of the adaptive immunity. Adaptive immunity. Adaptive immunity. Please, I want you to pay good and very attentive mindset towards what I'm teaching right now. I need your attention. I really need your understanding. I want you to give me all your ears so that we can understand this, because it's a little bit tricky, but when you understand the concept, you get there. So the first thing, it affects a macrophage. The macrophage has MHC1 and MHC2, which are able to express the viral peptide. So, but the macrophage is inactive. For it to be inactive, it needs a T helper cell. So I'm going to draw a T helper cell here. A T helper cell. So it's going to be T helper 0. It depends on the cytokine that will activate it to produce different types of T helper cell. So it needs a T helper cell to activate it before it can be able to activate a CD8 pre-cytotoxic T cell into a cytotoxic T cell that will do the killing. So what's now happening is we have a TH0, and do not forget, T helper cells can be TH1, TH2, T helper 2, Treg, TFH, and T17, TH17. So these are the 5 different types of T helper cells: TH1, TH2, Treg, TFH, and TH17. So depending on the cytokine that is released to differentiate the T helper 0 cell, depends on it, that can give you different types of TH1 to TH17. So now, let's say we want to produce a TH1, a T helper cell, a TH1. We'll call this my TH1. So now the TH0, do not forget, the T helper cell is a type of CD4 cell, so it's mediated by a CD4 cell. So now we want to activate our macrophage. We want to activate the macrophage. So the CD4. So we want to activate the macrophage. So I'm going to shift this here so that you see the diagram better. I'm going to shift it here. Okay. This is my green. Okay, let me do it this way. This is the macrophage. And I have macrophage. MHC2. And I'm going to bring my TH0. So I'm trying to produce a TH1 cell right now. Okay, there's a CD4 type of cell. So this will now be mediated by the T cell receptor, TCR, definitely mediated by CD4. you all know. Then on the body of the macrophage we have B7 and we have CD28. CD28. Another name for B7 is CD80 or CD86. Do not forget, this is a real point for those that are writing exams under immunology and viral infection. B7 is also called CD80 or CD86. So when all these things are bound, this is mediated by CD4, please. This is mediated by CD4. When all these things are bound, the macrophage will release interleukin 12. By so doing, it will differentiate the TH0 helper cell to TH1. So you need IL-12, interleukin 12, to differentiate the TH0 to T helper 1. Are we together? So this is it. Let's get a little recap. It is inactive. It has MHC1 and MHC2. It needs a T helper cell to be active. But for it to have a T helper cell, it needs to differentiate to different types of T helper cells, where we have TH1, TH2, Treg, TFH, and T17. TH17. Sorry. So now we want to have a TH1 helper, TH helper cell. So TH1 helper cell. So for it to do this, it needs to bind to different parts of the macrophage where it is mediated by the T cell receptor, CD4 precisely. Then we have CD28 binding to B7 on the macrophage's body, on the body of the macrophage, excuse me. So when these things are bound, the macrophage releases interleukin, a cytokine, interleukin 12, that differentiates the TH0 to TH1. So now, after doing this, the TH1 also does two things. First, it produces two different substances. The first one it produces is called interleukin 12, which is self-stimulatory. Why it's called self-stimulatory? The TH1 produces IL-12 because this enables it to undergo clonal expansion. Clonal expansion. So please take down these notes. When TH0 is differentiated to TH1, the TH1 also produces IL-12, which is a self-stimulatory substance that allows the TH1 to proliferate. That means to make different copies, enough copies that will do the killing, which is also called clonal expansion, cloning. It makes a lot of copies of it. Then it also produces interferon gamma. It produces IL-12, which is self-stimulatory, and also interferon gamma. I will tell you the function of interferon gamma later. On the body of the TH1, we have our CD40 ligand. CD40 ligand. We have our CD40 ligand. Excuse me. CD40 ligand. CD40 ligand. On the body of the macrophage, we have our CD40. It has always been there. I didn't just write it. Our CD40. Sorry, let me use a black pen again. CD40. When these two things are bound, it activates. CD40 now goes down and activates the macrophage. When it activates the macrophage tends to produce and express more B7 on the body. Please, I want you to understand this. I'm going to go back again. So TH0 differentiates to TH1 by the production of the IL-12 from the macrophage, which stimulates the TH1 to produce interferon gamma and IL-12, the same cytokine that was released from the macrophage, which is self-stimulatory, that make TH1 to proliferate and expand through clonal expansion. By so doing, on the body of the TH1 cell, you have CD40 ligand. The macrophage has CD40 on it. When these two things are bound, they allow the macrophage to continue to express B7 on the body. So this is what happens. On the CD8 cell, pre-cytotoxic T cell, this is really beautiful. I want you to pay serious attention to this. Okay, so I'm going to draw it here. So my CD8 pre-cytotoxic T cell, cytotoxic T cell. So this will be mediated. Now just watch this, please. This is your B7. So my MHC1, I'm going to bring it here. So it can be written anywhere. MHC1. I'm going to write it here. So the CD8 pre-cytotoxic T cell will definitely express CD28. CD28. Then this is mediated by T cell receptor. This is mediated by T cell receptor. Let me make this bigger so that we can have a clearer picture. Yes, we can have CD8 T cell, pre-cytotoxic T cell. So this is a T cell receptor, which is definitely mediated by CD8. So when all these things are bound, the CD8 binds the CD28 of the CD8 cell binds to the B7, which are expressed as a result of this bound between TH1 and the macrophage. So it allows the expression of B7 on the body of the inactive macrophage. When these things are bound, when the macrophage binds to the CD8 through the CD8 and the T cell receptor, it now activates the macrophage to release what again? Interleukin 12. Interleukin 12. This is really beautiful. So now let's take a general recap from the beginning. I told you the macrophage will now release interleukin 12 that will activate the CD8 T cell, pre-cytotoxic T cell, to become cytotoxic. Toxic means it's toxic to cell. Cyto means cell. Cytotoxic T cell that will now do the killing. So I'm going to take a recap. The virus infects a macrophage. When it infects the macrophage in this case is inactive. Since it's inactive, it expresses both MHC1 and MHC2. When it does that, it needs a T helper cell that will help in activating it so that it can produce IL-12. It can produce IL-12 that will activate the CD8 pre-cytotoxic T cell to become a cytotoxic T cell that will do the killing. So these are the processes. So now, when it's inactive, it needs a T helper cell. Before it is activated, the T helper cell uses the T cell receptor to bind to the MHC2. B7 is expressed on the body of the macrophage. We have CD28 here. CD4 is the mediator here. B7 is also called CD80 or CD86. So after these two things are bound, IL-12 is being released from the inactive macrophage that will stimulate the differentiation of T helper cell to TH1. What happens? TH1 now produces these two substances, your IL-12, which is a cytokine, and interferon gamma, which IL-12 becomes a self-stimulatory substance, meaning it allows TH1 to proliferate and undergo clonal expansion, making enough copies of TH1 cell. It has CD40 ligand. On the body of the TH1, which binds to CD40 that is able to activate the macrophage to allow the expression of enough and multiple B7 on the body. This attracts the attention of the CD pre-cytotoxic T cell, where CD8 now pre-cytotoxic T cell binds to the inactive macrophage so that it becomes active through CD8 T cell receptor B7 binding to CD28 and allows the production of IL-12 that will activate the pre-cytotoxic T cell to become cytotoxic. Thank you very much. So this is where we're going to stop. I hope you take a very, very attentive recap on this particular subject, and do not forget to like and hit the subscribe button. I know it might be tedious for you, but follow it bit by bit, you will get the picture. So after this video, we're going to talk about how does cytotoxic T cell do the killing, and how do we have memory in case we have an invading virus, exactly the same virus that infected us from the first place. Thank you very much.